The fossil record is like a time capsule offering us a rare glimpse into Earth's distant past. Though much of it remains fragmented, every so often an extraordinary discovery emerges, shedding new light on the biology of creatures that walked the planet millions of years ago. While dinosaur fossils are relatively common, most of what we uncover consists of hard parts, bones, teeth, and armored plates. These remains help scientists reconstruct skeletal structures, but they leave many unanswered questions about a dinosaur's soft tissue anatomy. Occasionally, we get lucky. Impressions of skin, scales, and even feathered plumage have been found, giving us a better idea of how dinosaurs looked in life. But there's an even rarer type of fossil, one that preserves internal organs. These finds are few and far between, yet they provide some of the most breathtaking insights into prehistoric life. Today, we're going to explore 3 such discoveries, fossils that contain preserved internal organs. 1. Asophenix, the fossilized baby dinosaur with a preserved digestive system. One of the most astonishing dinosaur fossils ever uncovered, belongs to a small, meditating theropod found in 113-million-year-old rock formations in Italy. This juvenile dinosaur, named Scipionyx samniticus, holds a unique distinction. It still contains the fossilized remnants of its internal organs. Inside its ribcage, paleontologists have identified what appears to be a heart, stomach, intestines, trachea, and other soft tissues, preserved after more than 100 million years. Scipionyx is truly one of a kind. No other dinosaur fossil has displayed such extensive internal organ preservation, making it one of the most groundbreaking discoveries in paleontology when it was officially described in 1998. Interestingly, Scipionyx was originally unearthed back in 1981 by an amateur fossil collector in a small quarry northeast of Naples, Italy. However, at the time, he didn't recognize the significance of his find. Believing it to be a prehistoric bird, he began preparing the fossil at home, carefully removing excess rock and even applying vinyl glue to reinforce the bones. In an attempt to make the fossil appear more complete, he even added an artificial tail made from extra rock slabs. It wasn't until 1993, after watching Jurassic Park, that he realized he might be in possession of something truly extraordinary. Taking the specimen to paleontologists at the Milan Natural History Museum, experts quickly confirmed that this was not just any fossil. It was the first Nanavian dinosaur ever discovered in Italy, between 1993 and its official naming in 1998. Museum experts carefully removed the added materials and prepared the fossil properly, revealing a stunningly preserved skeleton. But the real shock came when they discovered mineralized internal organs still intact within the dinosaur's body cavity, a truly unprecedented find. Measuring 237 millimeters, 9 inches, in preserved form, but estimated to have reached 461 millimeters, 18 inches, in life. This tiny theropod was likely no older than 3 weeks when it perished. Despite its diminutive size, the Scipionyx fossil provided an extraordinary glimpse into the soft tissue anatomy of dinosaurs. Initially classified as a Compsognathiid, a family of small predatory dinosaurs, including Compsognathus, recent studies have sparked debate over its true identity. Some researchers propose it may have been a hatchling or charada, while others even suggest a connection to What makes this fossil particularly remarkable is the preservation of not just impressions, but actual 3-dimensional internal organs. The internal organs of Scipionyx were preserved through a rare process where the original organic material was replaced by calcium phosphate minerals. This type of fossilization captures extraordinary detail, suggesting that this dinosaur was rapidly buried after its death, perhaps by a sudden flood or sediment collapse. One of the most fascinating features can be seen at the base of its neck. There, paleontologists identified 8 to 10 tiny C-shaped rings, partially embedded within fossilized connective tissue. These structures are evenly spaced and almost certainly tracheal rings, cartilaginous reinforcements that help maintain the shape and flexibility of the windpipe. In other words, we're looking at the preserved remains of a dinosaur's throat from over 113 million years ago. But the most striking of Scipionyx's internal organs are undoubtedly its which are preserved in astonishing 3-dimensional detail. One paleontologist even described them as lumpy and shiny, eerily similar to what you would see when dissecting a modern animal. It's mind-blowing to think that we can still observe the actual texture of this dinosaur's gut after all this time. Interestingly, The fossil revealed that the intestines were positioned much further forward in the body than scientists had previously assumed for non-avian dinosaurs. The level of detail is so extraordinary that researchers could distinguish different sections of the intestines, including the duodenum, jejunum, and ileum, terms typically reserved for describing living creatures. Following the intestines further along, paleontologists even located the rectum, and beyond that, an intact fecal pellet, or in simpler terms, fossilized poop. But it gets even better. Looking closely at the surfaces of the intestines and rectum, paleontologists discovered faint branching traces of blood vessels, remnants of the network that once carried oxygen and nutrients throughout the digestive system. This level of preservation is almost unheard of in dinosaur fossils. By carefully studying the remains, researchers were even able to reconstruct the order in which this baby dinosaur ate its final meals. First, it ate a fish. Then, it consumed a second fish. After that, it caught and swallowed a lizard. A second lizard was eaten next. Finally, it finished off either another lizard or another fish. This means that Scipionyx, despite being just a few weeks old, was already a skilled little hunter, preying on both fish and reptiles. This incredible discovery offers an unprecedented snapshot into the life of a dinosaur, not just its anatomy, but its behavior, diet, and even its digestive process. It's rare enough to find fossilized skeletons, but to have a glimpse into an individual's last meals, that's something truly remarkable. One of the most intriguing aspects of Scipionyx's fossilized stomach contents is what it suggests about its behavior, or, more specifically, its upbringing. Given that this hatchling was only a few weeks old, it's unlikely that it had the speed or skill to hunt fish and lizards on its own. Some paleontologists believe this could be evidence of parental care, meaning an adult may have provided food for the young dinosaur, much like modern birds and crocodiles do for their offspring, just in front of the intestines and stomach. Researchers noticed a reddish stain embedded in the surrounding sediment and bones. Its color and placement within the body suggest it may be the remains of a vital organ, likely the liver. Further analysis revealed that the stain contains iron-rich minerals, which likely originated from the breakdown of hemoglobin, the protein in red blood cells that carries oxygen. This means that, incredibly, we may be looking at the degraded remains of the dinosaur's actual blood, preserved for over 113 million years. But the liver might not be the only organ responsible for this iron-rich residue. In modern birds and crocodiles, the heart and liver are located close together, and since non-avian dinosaurs share anatomical similarities with their closest living relatives, it's possible that the heart was also present in this region. Another potential contributor, the spleen, which is responsible for storing blood and is also found near the liver in modern reptiles and birds. Even though no direct traces of the heart's muscle tissue remain, the fact that we might have fossilized blood from this dinosaur it's an incredible example of how soft tissues, normally lost to time, can sometimes leave behind chemical fingerprints that still tell their story millions of years later, as if this fossil wasn't already remarkable enough. Patches of three-dimensional muscle tissue have also been preserved, revealing an astonishing level of detail. In multiple areas of the fossil, researchers have identified bundles of muscle fibers, some of which even show the individual banding patterns seen in modern muscle tissue. When examined under a scanning electron microscope, these fine structures appeared to be the microscopic units that allow muscles to contract. Some of the best preserved muscle fibers were found at the base of the neck, a region crucial for movement and hunting. Another well-preserved patch of muscle is located at the base of the tail, again showing clear fiber structures. This level of detail allows paleontologists to study how this young theropod's muscles were arranged and functioned, giving us a rare glimpse into the strength and movement capabilities of dinosaurs in a way that bones alone could never provide. The Scipionyx fossil continues to reveal astonishing details, particularly in its musculature. In the pelvic and hindular region, at least 3 distinct muscles have been identified, including the primary muscle responsible for upper leg movement. This remarkable preservation allows paleontologists to study dinosaur locomotion with a level of precision never before seen. Another striking feature of the Scipionyx fossil is the preservation of horny sheaths on its claws. In life, these keratinous coverings extended the length of the talons, making them even longer than what fossilized bones alone suggest. This is an important reminder that dinosaurs' claws were often more formidable than their skeletal remains might indicate. The unprecedented level of detail in this fossil has been invaluable for understanding soft tissue anatomy in non-avian dinosaurs. The insights gained from this tiny theropod would have been impossible to gather from skeletons alone. Kedua Dormongwongai the raptor, with preserved organs, moving on to another incredible find. A raptor species even older than Scipionyx has provided another rare glimpse into the internal anatomy of dinosaurs, dating back to 121 million years ago. This fossil comes from northern China, where it was unearthed in near-perfect condition. In 2022, paleontologists officially described this remarkable specimen, naming it Dormongwongai. This small dromaeosaur, measuring about 1.5 meters, 4.9 feet, in length, was likely covered in a thick coat of feathers. Impressively, remnants of its plumage can still be seen along the head, neck, back, and tail, an exceptionally rare feature in the fossil record. Adding to the uniqueness of this find, a small frog skeleton was discovered right next to the raptor's foot, possibly indicating an interaction between the two animals. However, the most astonishing aspect of this fossil lies inside its body. A bluish-colored layer is visible in the lower abdominal region, which closely resembles the layout of the intestines preserved in extending forward in the same manner. This layer provides strong evidence that these dinosaurs had similar digestive anatomy. Upon closer examination using a scanning electron microscope, scientists found that this layer was composed of densely packed a feature identical to the preserved intestines of This discovery further supports the idea that some dinosaur species had highly structured and well-organized digestive systems, not unlike those of modern birds. The mineralization process was likely driven by decayed bacteria that became active shortly after the dinosaur's death, helping to preserve the tissue by replacing it with microscopic mineral crystals that perfectly replicated the shape and arrangement of its digestive system. Interestingly, just like in this suggests that the highly acidic environment of the stomach continued to break down tissue even after death, leaving no preserved traces of the organ. But perhaps the most intriguing aspect of this discovery is the similarity in intestinal structure and placement between Darlong and despite the two species being distantly related. If Scipionyx is indeed closely related to Carcharodontistris or Spinosaurids, while Darlong belongs to the Dromaeosaurids, then this suggests that carnivorous theropods may have had relatively conservative digestive systems. In other words, their gut structure remained largely unchanged across different lineages. This remarkable insight into dinosaur soft tissue biology would have been impossible to confirm without these exceptional fossils. So far, we've explored fossils that preserve internal organs, but there's another mind-blowing discovery, one that retains traces of an actual dinosaur brain. This is an extraordinary find because brain tissue is one of the most fragile parts of an animal's body. It breaks down rapidly after death, making it almost impossible to fossilize. Typically, when paleontologists study dinosaur brains, they rely on endocasts, natural or artificial molds of the brain cavity inside the skull. These provide a general shape of the brain, but lack soft tissue details. However, a one-of-a-kind fossil suggests that, in at least one rare case, actual brain tissue from a dinosaur was preserved. In most cases, scientists can only study the brain structure of dinosaurs by scanning the skull and reconstructing the empty cavity where the brain once sat. However, in this extraordinary fossil, actual brain tissue was discovered, attached to the surface of a dinosaur endoc. 3. Iguanodontian dinosaur, the astonishing discovery of a fossilized brain. This fossil, found along a beach on the southern coast of England, comes from a geological formation dating back 133 million years and belongs to an Iguanodontian dinosaur, a large, robust herbivore. The discovery of part of this ancient reptile's brain tissue has provided unprecedented insight into the physiology of these long-extinct creatures. At first glance, the fossil looks ordinary, just a brown pebble, but upon closer inspection, it reveals something astonishing. It is a partial brain endocast, a natural mold formed when sediment filled the empty space left behind by the brain after the animal's death. The most remarkable part, the surface of this endocast preserves mineralized remains of the brain's outer membranes, the layers that surrounded the brain during life. Even more incredibly, individual blood vessels can be seen in scanning electron microscope images, and researchers believe that some fragments of the outer cortical layer, the actual gray matter, may also be present. To help interpret these findings, Paleontologists compared this fossilized brain tissue with previous discoveries of more complete iguanodon brain endocasts. By examining the shape of the brain cavity, scientists could reconstruct what the brain likely looked like when it was still inside the skull. The blue section in the study represents an enlarged view of the expected brain anatomy. The Roman numerals indicate major cranial nerve canals that would have controlled various functions. The lighter blue area highlights the specific part of the brain structure preserved in the fossil. The green section shows the extent of the preserved outer brain tissue. The red sections indicate layers of a mineral called cytorite, which formed after the dinosaur's death. The blue markings correspond to fragments of bone. By carefully matching the features of the fossil with known brain structures, scientists confirmed that this fossil preserves real brain tissue, making it one of the most exceptional discoveries in the study of dinosaur neuroanatomy. These bones could have been part of the original brain case, or they might have come from another source and become mixed with the sediment that filled the empty space where the dinosaur's brain once was. It's likely that this iguanodontian died near a shallow, stagnant body of water, and after its death, the top of its head ended up buried in the sediment at the bottom. The water conditions were probably acidic and low in oxygen, effectively preserving the soft tissues before they could fully decompose, almost like a natural pickling process. However, the rest of the dinosaur's body was lost, leaving behind only this partial endocast of the brain. The outer membranes of the brain, known as the meninges, along with associated blood vessels, were preserved due to the rapid formation of calcium phosphate crystals, which replaced the original organic tissue. This process, called phosphorylation, is the same type of preservation seen in and it was only possible due to the unique chemical conditions of the water where the dinosaur died. By analyzing the fossil under a scanning electron microscope, scientists identified tiny mineralized fibers in the outermost brain membrane, labeled DM in the research. These fibers, arranged in a ribbon-like pattern, are believed to be phosphatized collagen bundles, the protein that forms the protective outer layers of the brain. In some parts of the fossil, a different texture was observed, resembling the arachnoid matter, another protective membrane of the brain. Interestingly, in modern crocodiles, the arachnoid matter directly overlays the brain itself, unlike in mammals, where additional protective layers exist. This fossil suggests that iguanodontians had a brain structure more similar to crocodiles than mammals. Even more astonishing, within the preserved arachnoid matter, there are faint impressions that appear to be the ridges and folds of the actual gray matter, meaning that this fossil provides an unprecedented glimpse into the structure of a dinosaur's brain. In some sections of the deeper layers of the endocast, scientists have identified highly intricate textures at a microscopic level. These areas lack the features typically associated with the protective brain membranes, which suggest that they might actually be mineralized fragments of gray matter, the cortical tissue of this dinosaur's brain. This possibility is nothing short of astonishing. The discovery of fossilized brain tissue raises fascinating questions about dinosaur intelligence. In modern reptiles, the brain usually occupies only about half of the cranial cavity, with the remaining space filled by blood vessels and synapses that help with blood drainage. Because of this, scientists have long assumed that dinosaur endocasts mainly represent the empty space around the brain, rather than the brain itself. But in this particular iguanodon brain fossil, the gray matter appears to be extremely close to the inner surface of the brain case, with less than a millimeter of space between the meningeal tissues and the brain itself. This raises the tempting possibility that we may have underestimated the brain size of dinosaurs. If their brains tightly filled their skull cavities, their intelligence might have been greater than previously assumed. However, scientists caution that this might be an artifact of the fossilization process. It's possible that as the brain decayed, it collapsed inward, pressing against the membranes, making it appear as though it took up more space than it actually did. As one of the paleontologists who studied the fossil explained, since the brain's lobes aren't clearly visible, it's impossible to say for certain how large this dinosaur's brain was. While it's intriguing to consider the idea that dinosaurs had larger brains than we thought, this particular specimen does not provide enough evidence to confirm it. What makes this find so exciting is the fact that a dinosaur brain, one of the most fragile and unlikely tissues to fossilize, managed to beat the odds and partially preserve. This gives paleontologists hope that other fossilized dinosaur brains could be out there, waiting to be found. Perhaps it's just a matter of time before we uncover a dinosaur brain that reveals even more about the internal structure and function of these prehistoric minds. And with that, we conclude this exploration of some of the most astonishing dinosaur fossils ever discovered. Click on the video on your screen to keep enjoying our content. See you in the next video.